Okay. Good morning, everyone. I'm Nelson Lau, the director of the Boston University Genome Science Institute, and it's my pleasure to welcome you to our 16th annual research symposium, where we bring together all the genetics and genomics researchers on both the Charles River campus and the medical campus here to present your work. And to put a little skin in the game, we've also made this as a competition where we're going to have 6 lightning talks between now and noon. And then from noon to 2, we will have a poster session. And in both the lightning talks and the poster sessions, there will be a competition for trainees to potentially win $10,000 from the GSI to support any kind of fanciful genomics project you would like to spend that money on. And when you decide to spend that money, I hope you will consider the great vendors we have here that will be running a vendor show between 12 and 2. I'd like to thank all these vendors here on our brochure and on our poster map and back there that will be describing their products for you to conduct your latest and greatest genomics research. I just want to remind you that we also have other events going on in the GSI. We have these seminars on November 25th with Bess Frost and December 10th with Michael Bain. And we encourage other students as well as PIs to participate in GSI seminars. If you have a candidate person you'd like to invite, let us know. My co-director, Dr. Grisha, can help you with all the logistics. I'd also like to thank Dr. Flynn for helping me organize the poster judging session, which is going to be, let's see, next slide here. So judges, the faculty, please scan the QR code and help judge the posters, because poster sessions also eligible for the poster award here. And we're also looking for thoughts on future GSI workshops in the spring. We've had a couple companies like Active Motif, Illumina, come give talks here. So we have a couple technologies we'd like to maybe encourage you to try out. This slide deck's just going to rotate around during the day. So if you miss something here during the poster session, during the lunch, that'll be at noon. Please check that out, seeing what's on here on these slide decks. And lastly, I'd like to make another plug for the Bioinformatics Master's Students Internship Program, which is going to be here. We have Dr. Adam Labradorf here, as well as Dimitri Leifer, who will be coming later today. If PIs here are looking for master's students from the bioinformatics program to help out with research projects, please reach out to Adam and Dimitri. We will run this program again in the spring, so please keep an eye out for this. I know the GSI sends you guys a lot of emails. Please check them out and keep attentive to all those things, because we try to establish a very vibrant genetics and genomics program here on the Med campus as well as the Charles River campus. And now that we have a new president, Dr. Gilliam, we hope to strengthen those ties between our two campuses. Without much further ado, I'm going to start off with the trainee lightning talks. We have 6 of them. They'll be 10 minutes each, 8 minutes long with 2 minutes questions. And I'd like to introduce Natalie Campbell from the Department of Anatomy and Neurobiology, and she'll be talking about oligodendrocyte development and myelination. Let's see, where is it here? All right, and you can either use this or this microphone here. Okay, yep, that's a good pointer.
Hi, everyone. I'm Natalie. I'm a third-year PhD candidate in the Zeldich Lab in the Department of Anatomy and Neurobiology. And today I'll be talking about assessing oligodendrocyte development and myelination in Down syndrome using human forebrain assembloids.
You might need to use this microphone here.
Testing?
Yeah.
Okay, great. So Down syndrome was first characterized by John Langdon Down in 1862, and then about 100 years later, Dr. Jerome Lejeune discovered the genetic cause, which is the triplication of the 21st chromosome. Down syndrome is characterized by developmental changes over time in both gray matter and white matter. In gray matter, we observe decreased neurogenesis, decreased brain size, larger ventricles, microcephaly. And then in white matter, we see delayed and diminished myelination. And features of this myelin include thinner myelin sheaths, fewer myelinated axons, leading to reduced conduction velocity. In Down syndrome, we would expect that there would be a triplication of the genes on the 21st chromosome, but we also observe global transcriptome dysregulation. There are up and down regulated genes on every chromosome, not just the 21st as we would expect. In this study that discovered this global transcriptome dysregulation, they also found a module of genes enriched for myelination, and differences between euploid and trisomic for these genes excuse me, increased over neocortical development and coincided with the onset of myelination. Oligodendrocyte progenitor cells, or OPCs. Oligodendrocytes are the cells that produce myelin, and oligodendrocyte progenitor cells migrate through the telencephalon during development. They start ventrally in the subpallium, the first two waves of OPC migration, and the third occurs dorsally in the pallium. To model oligodendrocyte development in this way, we use induced pluripotent stem cell-derived cortical organoids from isogenic paired lines. Isogenic means that they are genetically identical aside from the triplication of the 21st chromosome. So through this, we can develop our cortical organoids, and we have ventral and dorsal organoids to mimic the brain regions that we're trying to model, the subpallium and the pallium. And then in order to create a more physiologically relevant model of a brain-like environment, we fuse these organoids into forebrain assembloids. So you can see the assembloids like in the tube there. So these are what our assembloids look like at day 150. By day 150, we would expect our oligodendrocytes to be able to myelinate and or to be mature and to produce myelin. Here we can see our, can you see the, here we see our dorsal section here and our ventral section here. And we know that the oligodendrocytes are migrating from this ventral section into the dorsal section. So here we have frozen or fixed our assembloids at day 150, frozen them into blocks, sectioned them, and performed immunohistochemistry to detect markers of myelination. So CC1 is a pan-oligodendrocyte marker that is expressed in both immature and mature oligodendrocytes. We have MBP, or myelin basic protein, which is expressed prior to and during myelination, and BCAS1, which labels actively myelinating oligodendrocytes. So when we look between our euploid and trisomic assembloids at day 150, we don't see any change in the amount of CC1 positive cells, meaning that our oligodendrocytes are being produced to the same extent in both euploid and trisomic. We also see that when we look at co-expression of CC1 and MBP, that there's a reduction in our trisomic assembloids, and this would be in line with previous studies that show there's less MBP in trisomic, or in Down syndrome, excuse me. However, what was surprising to us is that when we looked at the co-expression of CC1 and BCAS1, which are those actively myelinating oligodendrocytes, we see an increase in trisomic. And this is a discrepancy in the developmental trajectory, because we would expect our oligodendrocytes to mature and then begin to myelinate. However, it looks like we have premature recruitment of our oligodendrocytes for myelination, while the expression of MBP is diminished. So in order to look more closely at the genes associated with these processes, we did single cell RNA sequencing, and we identified, or in all three lines that we have 3 Down syndrome lines, 2 female and 1 male. So we did sequencing on our day 150 assembloids, and we were able to discover multiple cell types. We have excitatory neurons, inhibitory neurons, glial cells, and progenitors. And specifically, we wanted to focus on our 2 clusters of oligodendrocyte progenitor cells. So we dug a little deeper into the differentially expressed genes in these 2 clusters alone. So when we looked at these genes, we ran a David analysis to discover which biological processes are being dysregulated in trisomy. And we discovered Go terms related mostly to neurons and synapses, things like synapse itself, post and presynaptic membrane, dendrites and axons. So then we had to look and see, do we see these patterns in the literature? Is oligodendrocyte development and myelination activity dependent? And we found some evidence to support this as well. So in order to confirm these phenotypes functionally, we needed to do another method. And so we used calcium imaging to assess the functional characteristics of our neurons. We used a GCamp8S calcium indicator under the synapsin promoter to label them. And when calcium ions enter the cell, the GCamp fluoresces, and we are able to detect neuronal activity through this. And as you can see in our videos, in the euploid, the cells are firing not synchronously and much more frequently than in our trisomic. And we can see like a great burst of firing all together in the trisomic. So our preliminary results from just 5 euploid and 5 trisomic assembloids reveal this hypersynchrony in our trisomic organoids or assembloids, my bad, and this reduced frequency of firing. So overall, we think that this abnormal neuronal activity is leading to changes in myelin, and that it's driven by the genes that are dysregulated in these OPC clusters specifically. So in conclusion, we're able to model myelin deficiencies in Down syndrome with our trisomic assembloids. Excuse me. While there's no difference in the generation of oligodendrocytes, we do see discrepancies in markers of maturity and active myelination in trisomy. We can see that transcriptomic studies reveal dysregulation in the expression of genes related to neuronal activity, specifically in our OPC clusters. And we see hypersynchronous neuronal activity in our trisomic assembloids, which can be causatively linked to dysregulation in our oligodendrocyte biology. So in the future, I hope to, I've already begun to explore the composition of myelin in Down syndrome organoids using electron microscopy, and I've been able to locate compact myelin in our euploid organoids. So I hope to continue this project. We also want to identify the transcriptomic signature of the abnormal oligodendrocyte to neuron communication that we're observing. And then furthermore, we want to determine the impact of the epigenetic landscape in trisomy 21 and how that's related to this abnormal oligodendrocyte neuron communication. So I'd like to thank my lab and all of our collaborators, as well as GSI, for allowing me to be here today. And I'm willing to take any questions. Thank you.
Great. Thank you, Natalie. Any questions from the audience?
Thanks. That was a great talk. I might have missed this, but how are you generating your euchromatic lines?
Oh, so we have paired isogenic induced pluripotent stem cell lines. So we get them already paired. They are generated through like the cloning, in the cloning process. Sometimes they lose the extra chromosome, and that's how we get the identical euploid.
Okay, any other questions? Okay, great. Thank you, Natalie. Our next speaker is Jenna Libera from Department of Pharmacology, Physiology, and Biophysics. And let me get your talk up. This one? All right. Thank you. I might have to use the microphone. The microphone here.
This one?
Oh, that
This one.
Yeah. And then push that. Yep, that's advanced. And then the little laser pointer's on that thing too.
Great. Everyone can hear me? Okay. Hi, everyone. My name is Jenna Libera. I'm a fifth-year PhD candidate in the Wallison Lab, and today I'm going to be sharing some of my data looking at N6-methyladenosine, or M6A, and Alzheimer's disease. So N6-methyladenosine, or M6A, is the most common post-transcriptional modification that occurs on RNA, and it regulates RNA metabolism. So when you think about proteins regulated by PTMs like phosphorylation, this is very analogous to what happens at an RNA level. M6A modifications are very dynamic, and they are installed by methyltransferases called writers, and they're simultaneously deinstalled by demethylases called erasers. And what really drives the biology or the biological function of M6A sites is by RNA binding proteins called readers. So readers are comprised of many proteins that have what's called a comprised of a YTH domain. And this is very specific to the M6A modification that allows the protein to bind and recognize this residue along the RNA transcript. And when you see what types of functions are implied when you have M6A modifications, there's many different processes of RNA metabolism that can be inferred by the binding of these many different proteins. Now, what's very interesting is that we study Alzheimer's disease in our lab, and many of these pathways are affected aberrantly in Alzheimer's disease. But there has never really been a study that looked at the specific residues of M6A and how that regulates these pathways and how that could be disrupted in Alzheimer's disease. So the goal of our research is really to identify the specific residues that are M6A modified and get a global feel for how the transcript level might be changing its methylation state, and then infer how that could be linked to these processes that are impaired in Alzheimer's disease. So in order to do that, I implemented a technique in the lab called DART-seq, which stands for deamination adjacent to RNA targets. This technique was developed by a woman named Kate Meyer at Duke University, and it has never before been studied in the context of Alzheimer's disease in postmortem tissue. And the way that it works is that we take our DART protein, which is a chimeric protein comprised of YTH protein with ApoBec deaminating enzyme, incubate that with RNA, and you're able to then kind of bind the M6A modified residues through the YTH domain, and simultaneously ApoBec converts cytosines to uracils. And so through reverse transcription, you then infer down here, whenever you have a thymidine adjacent to an adenosine instead of a cytosine, you then infer that that site was modified because these sites occur in a GAC or AAC motif. And this method kind of triumphs over other classical methods that are used in the field to study M6A biology. And so we're really excited that we're one of the first people to start applying this to Alzheimer's disease. And we use the core to do the DART-seq and the RNA-seq, which is really important for us. When we looked at our profile of what sites are changing, we saw that the largest domain of M6A modified sites occurs in the coding sequence, followed by 3' UTR. We also saw that there's that GAC or AAC enrichment motif, which means that we're kind of hitting the right residues when we're doing our method. And then when we looked at KEGG pathways to see which genes these sites are falling on, we were very happy to see that among the top ones are involved in RNA metabolism itself, such as RNA surveillance, splicing, nucleocytoplasmic export, etc. And of course, we also see many synaptic terms and neurodegenerative terms, including Alzheimer's disease. When we started to look at the data parsing out between our control and Alzheimer's cohorts, we used 19 control and 19 AD respectively, we saw a very interesting trend in the controls where there's this age-associated increase in M6A modified sites, which is lost in Alzheimer's disease, very interestingly enough. And when we looked at whether this pattern is holding across all of these different domains that we looked at, we saw that in the 3' UTR region, there's the most significant change that recapitulates this pattern, suggesting that the M6A sites specifically in the 3' UTR may be driving some of this disease pathology. When we performed a KEGG pathway analysis of the 3' UTR M6A sites, we saw that the GABAergic synapse was the top most enriched pathway, followed by many other growth signaling components and other synaptic terms like glutamatergic and dopaminergic. We then wanted to look at what differentially methylated transcripts were in our study. And funny enough, of course, we see that there's many GABA-related receptors. So this family might be co-regulated at the M6A modified level. We were also happy to see that APP, MAPT, and BACE1, which are all Alzheimer's-related genes, were also hypomethylated. And of course, you can see that there's many more hypomethylated genes in Alzheimer's disease compared to hypermethylated. When we overlapped our RNA-seq data with our DART data for methylation, we wanted to try to ask the question whether or not expression and methylation might be co-regulated, because that's an RNA metabolic process that we're interested in. And we saw that only 22 genes actually overlapped, but of course we saw GABRA1. And so that led us to kind of use this as a target to start looking at our biology more in Alzheimer's disease. And when we looked at GABRA1 3' UTR sites, we saw that this pattern was recapitulated in the global figure that I just showed previously, where you see an increase in GABRA1 sites across aging and controls, and that pattern is again lost in Alzheimer's disease. Now what we did was we took the individual patient, whether it was a control or an AD, and we wanted to find out whether there's a correlation at the individual level where expression and methylation might be regulating or associated with each other. And in our control patients, we see that there's a budding negative trend, but the p-value is not significant. But interestingly enough, when you go to the Alzheimer's cases, you see a very strong correlation, a negative correlation and a significant p-value. So this is suggesting that in the disease-specific cases, there's a negative regulation that could be happening where if a patient has higher M6A levels, it means that it's associated with lower transcript levels of the GABRA1 receptor. And this was really interesting to us because, as I mentioned, we see many synaptic terms that are being regulated. And so what we developed in the lab was a method to be able to isolate synaptosomes from our homogenate input, which is HOM versus SYN, and then look at this to compare between our control and AD cases. So in C, we validated the GABRA1 protein, of course, being more synaptically enriched. But in D, you could see that there's a quantitative significant difference between the healthy cases and the Alzheimer's cases in terms of GABRA1 protein in the synapse, with, interestingly, no change in the homogenate. And this is really interesting because, of course, in Alzheimer's disease, it's a neurodegenerative disease. So you think about synaptic loss, and maybe that could be a driver of this. But in ENF, we wanted to look at other terms that might be correlated with this data. So we looked at methylation, expression, protein in the homogenate, and protein in the synaptosomes. And interestingly, what we see for our healthy patients is that more methylation is associated positively with more GABRA protein in the synaptosomes with no correlation in the homogenate. However, we see a very interesting opposite effect in the Alzheimer's cases, where more methylation is associated with more protein in the homogenate with no correlation in the synapse. So this is an example of something we'd like to dive further into because we did not do sequencing on our synaptosomes yet. And so that's our immediate next step that we would like to undertake. So we could start comparing the synaptic methylation levels with synaptic transcript levels to kind of get more of a purview of how these processes might be affected in Alzheimer's disease. So in conclusion, I would like to recap what I've just shared with you. We used DART-seq to we implemented DART-seq to find methylation profiles in our human postmortem brains. We saw many more hypomethylated transcripts in Alzheimer's disease, many of which are related to synaptic functions. We saw that GABRA1 methylation and expression are negatively associated in AD, and that the methylation and protein levels might be opposingly correlated between control and AD cases, suggesting that there's disease-related differences in either synaptic translation or maybe localization, which of course are components of RNA life cycle and RNA metabolism that we're interested in studying. And so, as I mentioned, our next immediate steps would be to isolate synaptic RNA to confirm methylation and localization levels. And then in the future, we hope to explore genetic tools that might allow us to modulate these sites either in an in vivo or in vitro model, where we can then change the methylation profile of specific sites using a DCAS approach that then might be able to be recapitulated in these live models, essentially. And with that, I'd like to thank my whole lab for supporting me today, many mentors, the GSI, my DAC, and our funding sources. Thank you very much.
Thank you, Jenna. That was great. Questions from the audience? Yes, Hui Ping.
Thank you, Jenna, for a very interesting talk. So my question is that, so for the DART sequencing analysis method, so how much RNA you need to be used, and what's the quality of the RNA samples? Because you use postmortem brain tissue samples, and usually the RNA quality is not that good. There might be some degradation. Thank you.
I don't think I have RIN here. Our RIN averages 7.4 and 7.1 between our control and AD cases. The PMI on it is about 2 to 4 hours. So we got our brains from the Kentucky Brain Bank, and they have pretty low postmortem intervals. So the quality of our RNA was at least 7. And in terms of input, we used 50 nanograms of input, which is quite low for a lot of these studies. And we have to use a low input library prep kit to be compatible with this. But this is an advantage for using the DART-seq because many other methods like ME-RIP-seq, like methylated RNA sequencing, uses at least 1 microgram. So we're able to kind of get over that hurdle by using a lower input method. Right. Yeah, and it's great because the human samples are so precious, so we don't want to use as much. Thank you.
Thank you, Jenna. Fantastic.
Thank you.
Just to keep on time, we're going to move on to our third speaker, Sam Cranston from the CREM, talking about direct conversion of centenarian PBMCs. Let's bring up your talk here. Here you go. All right, here you go. Thank you, Sam.
Hi, can you guys hear me?
Turn
Oh,
it
it's
back on. Yeah. And there's the laser pointer. Sorry.
Thank you. Hi, my name is Sam Cranston, and I'm a trainee in Dr. George Murphy's lab at the Center for Regenerative Medicine. And today I'm going to be presenting for you my quest for the 100-year-old neuron, direct conversion of centenarian PBMCs as an in vitro model of aging and resilience. So our lab recently underwent a major shift in focus. We've shifted from studying blood and the hematopoietic system to now studying human aging biology. So what's the reason for this shift in focus? Why study aging in general? This chart illustrates the mortality risk associated with various significant diseases across age groups. As you can see, the risk of death from conditions like cancer, heart disease, and stroke increase dramatically with age. Aging is the primary risk factor for all the diseases you see here. So by targeting the aging process directly, we aim to delay the onset of all these diseases simultaneously. This approach could increase the years our population lives in good health and decrease the burden of aging-related disease on the healthcare system. So you might be wondering from my title slide, what is direct conversion? So this is Waddington's diagram, something you might be familiar with. And it has been used to show that normal mammalian development is unidirectional. You start with this embryonic stem cell, and that further differentiates into all the different cell types in the body. However, with the discovery of the Yamanaka factors in 2006, we saw that this process could be reversed in vitro via the reprogramming of somatic cells back to an induced pluripotent state, the induced pluripotent stem cell. These cells can then be differentiated into all the cell types across the body. So direct conversion, in comparison, is this idea that you could take a terminally differentiated cell type, like a fibroblast, and directly transition it into another cell type, bypassing the need for this pluripotent intermediate. So by bypassing this intermediate, it's been shown that directly converted cells actually retain the aging signature of the donor from which they're derived. Whereas an iPS cell is fetal in nature, these direct converted cells retain the age of the donor from which they're derived. So as I've kind of been hinting at, we've become very interested in developing these in vitro models of aging and resilience. We collaborate with the New England Centenarian Study and have access to hundreds of blood samples from centenarians, or people who live past 100 years old and display this exceptional resistance and resilience to aging-related disease. So we found that these individuals share protective molecular profiles that can regulate stress response and promote resilience. We believe that centenarians can provide a blueprint for living longer and healthier lives. So far, we've developed a bank of induced pluripotent stem cells derived from centenarians. This bank has acted as a renewable source of biomaterials from these rare individuals, and we've been using it for molecular profiling and functional testing of the pathways that promote resilience in centenarians. So we're now looking into direct conversion protocols as a complement to this IPS model. As I mentioned, these DC neurons would retain the aging signature of a centenarian, providing the exciting new opportunity to study 100-year-old neurons in the dish. So direct conversion protocols have historically focused on using fibroblasts or skin cells as a starting material. However, as I've been saying, we have this bank of PBMCs from centenarians. So we needed to develop a novel protocol for the direct conversion of peripheral blood cells into induced neurons to investigate the aging signatures of induced neurons derived from centenarians. So how have we been going about this? Through months of trial and error, we've refined this protocol for the direct conversion of PBMCs into induced neurons. We first isolate PBMCs from centenarians, and then we utilize a lentivirus to induce the expression of neuronal genes into these blood cells. Importantly, we can select for cells that have received this virus through the antibiotic pyrimycin. So now we have these blood cells that can express neuronal genes. We then plate these cells on top of primary human astrocytes. These astrocytes provide an adhesive substrate for blood cells to adhere to the dish and then go through the direct conversion process. The astrocytes also support neuronal growth and functionality as they would inside your brain. We then pattern our cells in this optimized neuronal medium and add in pyrimycin to remove contaminating astrocytes and non-converted PBMCs, leaving us with a purified induced neuron culture. So these are the results of this protocol. The red you see here is GFAP, a classic marker of astrocytes. And then the green, the exciting part, is MAP2, a hallmark neuronal marker expressed by all lineages of neurons. So you can see this clear MAP2 positive neuronal morphology separate from the astrocytes, demonstrating the successful direct conversion of peripheral blood cells into induced neurons. Furthermore, these images are taken from blood cells taken from a centenarian, representing the first successful direct conversion of primary centenarian cells. So we initially questioned if this would even be possible. However, these images show that it is. So this is a really exciting day for me and the rest of the Murphy lab, and the results of months of brainstorming and hard work. So now the question becomes, we have this population of induced neurons co-cultured with astrocytes and other PBMCs that haven't gone through the conversion process. So how do we isolate these cells? As I mentioned earlier, we've been able to utilize pyrimycin, the pyrimycin resistance in our induced neurons, to remove these contaminating cells and create a purified population of induced neurons. These images show a purified induced neuron culture after 7 days of culture in pyrimycin. So this experiment was only run a few weeks ago, and we're still awaiting the results of this analysis on the purified induced neuron culture. However, we expect these cells to retain the aging signature of the centenarian, providing an exciting new opportunity to study 100-year-old neurons in the dish. So this is our ultimate vision. We aim to have both our iPSC and direct conversion models working in tandem, using starting material from the same subject. We can currently make iPSC-derived neurons from the same primary blood samples that we are currently using in direct conversion. From iPS neurons, we can test that these cells may respond to the application of stressors and identify factors that promote resilience. From the opposite end of the spectrum, we could test rejuvenative interventions on our DC neurons aimed at decreasing the biological age and restoring functionality of these cells. In this way, we can generate a complete picture of how an individual may respond to an aging-related intervention, resulting in a highly specific and personalized model of aging and aging-related disease. Thank you for your time, and thank you to my mentor, Dr. George Murphy, and the GSI for inviting me to speak today.
Thank you, Sam. Sorry for those alarms there. Really great talk. I have one quick question. Have you compared the rejuvenation time between young PBMCs and the centenarian PBMCs?
Yeah, so that's the ultimate goal, right? And we've very, very recently developed this protocol. So Now it's about optimizing it and doing the sort of testing that you're alluding to.
Okay. Any other questions, Ben?
It's great stuff, Sam. Neurons come in many different flavors. So what type of neurons do you generate through the direct conversion? How does that compare to the type you generate through the iPSC conversion?
So the IPS neurons are cortical neurons. Again, very new protocol. We're still characterizing these cells, and we're awaiting the results of analysis on these cells that would help us further understand what type of neuron we're generating and the aging signatures that these neurons retain.
Awesome talk, fascinating. I would like to ask about the astrocytes. Astrocytes currently in your model provide support for the differentiation of induced neurons. And probably it's somewhere on your list to differentiate astrocytes and generate induced astrocytes from the same people. And if you do this, what the effect would you expect? Or if you have in the same culture the induced astrocytes together with induced neurons from the same individuals.
So you're saying like direct conversion of blood cells into astrocytes as opposed to neurons?
Yeah.
I'm sure, I think there is protocols for the direct conversion to develop astrocytes, but again, very new protocol. That's something we would be interested in looking at in the future.
All right. Thank you, Sam. Let's move on to our fourth speaker, Simon Liu from Department of Computational Biomedicine in the Data Science Core. Is your presentation here? Okay. All right. I'll let you talk about your title. Thank you.
Hello. Hello.
You turn it.
Hello. Oh. Hello. My name is Simon Liu. I am a bioinformatics assistant from the Chao Zhang Lab in the Computational Biomedicine Department and Data Science Core at BU. My talk today is titled "Transcription Factor ZB2 Determines the Cell Fate of Kidney Stromal Progenitors, and Loss of ZB2 Protects Mice from Proteinuria." This project is in collaboration with Dr. Sudhir Kumar from the Nephrology Department at BU. So starting off with some background information, ZB2 is a transcription factor that plays a crucial role in the growth and development of many organs and tissues before birth. It interacts with activated SMADs as a co-repressor through the SMAD binding domain and regulates cell differentiation and their fate during embryogenesis. Mutations in the ZB2 gene cause Mowat-Wilson syndrome, a rare genetic disease with multiple congenital defects, including kidney fibrosis. Patients with Mowat-Wilson syndrome develop kidney and urinary tract anomalies. So in a previous study by Dr. Kumar, it was found that ZB2 is expressed in kidney stromal progenitors and differentiated kidney stromal cells. We can see here the immunostaining results showing the co-localization of ZB2 with FOXD1, a stromal progenitor marker, and PDGFR beta, a parasite and fibroblast marker, which are two common differentiated kidney stromal cells. It was also found that loss of ZB2 in FOXD1 positive stromal progenitors leads to renal failure and early mortality. In mice where ZB2 was conditionally knocked out, the lifespan was significantly shorter, the overall body and kidney size was smaller, and the blood urea nitrogen levels were nearly fourfold increased when compared to their wild-type littermates. All of this is indication of impaired kidney function and kidney failure. So in conclusion from the study, it was determined that ZB2 controls stromal progenitor differentiation, and loss of ZB2 causes kidney fibrosis. In the figure on the right, we show that FOXD1 positive kidney stromal progenitors in wild-type mice where ZB2 is regularly expressed, FOXD1 positive stromal progenitors differentiate normally to fibroblasts and parasites. This leads to normal kidney stromal cells and microvasculature, as well as normal nephrogenesis, resulting in normal kidney function. On the other hand, where ZB2 is conditionally knocked out and absent, these FOXD1 positive stromal progenitors differentiate abnormally to myofibroblasts. This leads to kidney fibrosis and microvascular rarefaction, as well as abnormal nephrogenesis, resulting in kidney failure. Despite all this, however, it was found that ZB2 knockout mice do not develop proteinuria, where elevated protein is found in the urine. This is a common indicator of kidney impairment. So this suggests to us that even though ultimately kidney failure does occur in the absence of ZB2, the kidney's filtration processes are all preserved, preventing any leakage of proteins into the urine. So in order to investigate this, single-cell RNA sequencing was performed using 10x genomics chromium single-cell transcriptional profiling of kidney tissue in 2 wild-type and 2 ZB2 conditional knockout mice. The single-cell RNA seq analysis was performed using the SURAP package. So in our pre-processing, we use traditional methods to minimize noise and maximize significant gene expression. Starting with QC filtering, we filtered out cells with mitochondria percent greater than 50% and genes with 0 counts in all samples. We then performed normalization and identified variable features. 2,000 variable genes were selected for subsequent analysis. We integrated our 4 data samples together using SURAT integration to remove batch effects. And then we performed scaling and principal component analysis where 50 significant principal components were chosen. So in our clustering, we identified 10 overall cell clusters, and with the help of Dr. Kumar and his team, we annotated these clusters with the following cell types. And in particular, in relation to our hypothesis regarding preservation of filtration processes in ZB2 conditional knockout mouse kidney, our single-cell analysis found that podocyte number was increased in ZB2 CKO kidney when compared to wild-type mice. These podocytes are highly specialized cells of the kidney that contribute to the filtration ability of a kidney. They are terminally differentiated cells, so they do not replicate after development. And a podocyte number has a positive correlation with kidney health. A loss in podocytes in abnormal and injured kidneys will lead to renal impairment and ultimately kidney failure. So all this supported our previous hypothesis that ZB2 CKO mice do not develop proteinuria, and this could be the reason why. Oh, sorry. Wait. To investigate this further, we performed differential expression analysis and found that podocyte master regulator, WT1, was significantly upregulated in ZB2 CKO mice. In the volcano plot on the left, we see that WT1 is significantly upregulated, and on the right, the bar plot shows that WT1 positive cells are significantly increased in ZB2 CKO when compared to wild type. So in addition to this, we found that there is a significant upregulation of podocyte-specific marker gene expression in our vascular cell cluster of the ZB2 CKO mouse kidney. So somehow the vascular cell cluster was significantly expressing these podocyte-specific genes, which suggests a potential differentiation between vascular cells and podocytes in the absence of ZB2. The increased number of podocytes could be derived from its surrounding vascular cells. So we wanted to investigate this further, and to do that, we subset our podocyte and vascular cell clusters together separately from the rest of the data set, and we performed monocle 3 trajectory inference of the combined clusters. Through this algorithm, a learned trajectory was found to exist between podocytes and vascular clusters, further supporting our new hypothesis that derived podocyte cells are due to vascular cell differentiation. So, in order to validate this, Dr. Kumar and his team performed immunostaining results of WT1 and found that it is indeed expressed ectopically in interstitial cells in ZB2 CKO mice. This matches our analysis that shows that WT1 positive cells are significantly increased in both podocytes and vascular cell clusters in our single cell data. In addition to that, another podocyte marker, TLE4, was also found to be upregulated in the interstitial cells of ZB2 CKO mouse kidney. This is also shown in our single cell data. And then in close association with podocytes, parietal epithelial cells were found to not develop properly in ZB2 CKO mouse kidney. These parietal epithelial cells also play an important role in the filtration processes of the kidney. So as shown by the Claudin-1 marker gene, Claudin-1 positive cells were significantly increased in the ZB2 CKO sample. So these findings all support our hypothesis that kidney filtration function is preserved in the absence of ZB2. So in summary, ZB2 is expressed in kidney stromal progenitors and differentiated kidney stromal cells. Through our analysis, we found that podocytes and parietal epithelial cell marker genes are increased in ZB2 CKO mice. And then this led us to the hypothesis that FOXD1 positive stromal progenitor cells differentiate into podocyte-like and parietal epithelial-like cells in ZB2 CKO mouse kidney. And then this was validated, showing that podocyte protein WT1 and parietal epithelial protein Claudin-1 are ectopically expressed in the interstitial cells in the ZB2 CKO mouse kidney. In conclusion, we claim that stromal progenitors in developing kidneys may differentiate into parietal epithelial and podocyte-like cells without ZB2, which strengthens the kidney glomerular filtration barrier and protects mice from proteinuria. I would like to acknowledge the Chao Zhang Lab and our collaborators. Special thanks to Dr. Chao Zhang and Dr. Sudhir Kumar for their guidance and support. And also thank you to the Boston University Single Cell Sequencing and Confocal Core. Thank you.
Thank you, Simon. Any questions from the audience?
I have one question about the analysis. Why did you choose the mitochondrial filter to be too high as 50%? Like it's usually around 15 or 10%. Why did you choose 50%? That's way too high.
Sorry, can you repeat that?
Why did you choose the mitochondrial filter to be way too high as 50%? Like it's generally around 10% and not 50%.
I think this was a benchmark because we see in our graph that there aren't that many cells with mitochondria percentage greater than 50%. So you kind of just wanted to subset our data containing only relevant information. We didn't want to associate anything with mitochondria.
I noticed, so I think your goal was like to find out why the humans get disease, right? So the in your volcano plot, it looked to me like you had a lot of SLCs, which were down-regulated, which are usually transporters that I think are important. I don't know, but could be important for kidney function. Is that, are those things kind of, those SLCs, are those relevant to kidney function, and could those contribute to the disease process?
So I don't have that expertise in kind of the kidney genes, but the overall goal of the project was to see kind of why proteinuria wasn't developed in these diseased mice, even though they ultimately have kidney failure. Why were these processes protected? And we saw that it was due to podocyte gene upregulation as well as podocyte count increase. And the volcano plot showed that through the WT1 gene that was significantly upregulated.
Right, but my point is that underneath the general protection, you still might see signatures of kidney dysfunction, which could be in I don't know, kidney biology, the gene specifically, but could be in those SLCs, which are used to transport, like a lot of times, ions like sodium and potassium, things like that.
Yeah, I think that'd be interesting to investigate further.
Okay, thank you, Simon.
Thank you.
Okay, just in the interest of time, we're going to move on to our fifth speaker, Maria Valadas Ingersoll from the Department of Biology in the Davies Lab, talking about single-cell sequencing in coral. Should be really interesting to hear. This one. Great, thank you, Maria.
Hi, everyone. Thank you for the introduction. My name is Maria Valadas Ingersoll. As Nelson said, I am in the Department of Biology on the CRC. I'm a fifth-year PhD candidate in the Davies and Gilmore labs. And today I'm going to be taking us in a slightly different direction, but with some influence from the previous presentations on single-cell RNA sequencing and how it reveals immune system compartmentalization under symbiosis in the stony coral Oculina arbuscula. All right, so first I'm going to give you an introduction to coral. So coral belongs to the phylum Cnidaria. Cnidaria also encompasses jellyfish and sea anemones. And we know about coral a lot from environmental work. This is a coral reef in Panama. And if you look at the coral kind of from an outside perspective, it looks like this big rock structure. But if you zoom in, the coral is actually comprised of all of these clonal polyps. And if you zoom in further onto these clonal polyps, they have multiple tissue layers. In the most internal tissue layer, the gastrodermis, inside of those gastrodermal cells live photosynthetic algae. And these algae perform photosynthesis and transfer some of those fixed carbon sugars to the coral host, which the coral host uses. And the coral, in return, gives CO2 and nitrogens and phosphoruses to the algae. We know a lot about coral, and you've probably heard about coral in reference to coral bleaching. And what coral bleaching is a dysbiosis or a breakdown of that symbiosis. So under healthy conditions, you have a nice coral hosting its photosynthetic algae, and that's what makes it this like beautiful brown orangey color on the reef. But in stress conditions, like thermal stress is the most common one, the algae leave or are expelled from the host tissue. And this results in a white phenotype, or what we call bleached. And so this is what we see on the reef. And in tropical corals, this symbiosis is obligate, meaning that when the algae is not inside of the host cells, the coral will eventually starve and die. And this results in really devastation, a lot of devastation in the reef ecosystem. And so in my labs, I am really interested in answering this question of how is this cnidarian algal symbiosis established and maintained? And this is especially important in reference to how is that algae allowed to live inside of the coral cells without being destroyed by the host immune system? And in order to really study these different states of symbiosis, we use facultatively symbiotic models. And facultatively symbiotic models can exist in that dysbiotic or bleached, or what we call aposymbiotic state, or that symbiotic state perfectly healthily, because when they're aposymbiotic or bleached, they can buffer nutrient loss through heterotrophy. And we use two models predominantly in my research. I use this stony coral model, Oculina arbuscula, which you'll be hearing a lot more about, and this sea anemone model, Exaptasia pallida or Aptasia. And another big interest in our lab is the transcription factor, nuclear factor kappa B, or NF-kappa B. And what we've noticed in previous research done in the lab is that when we have a symbiotic, this is Aptasia, symbiotic Aptasia versus aposymbiotic Aptasia, in the symbiotic condition, there's a lower levels of this protein NF-kappa B. And NF-kappa B is a really important protein in the innate immune system in organisms from basal metazoa all the way to humans. And so what this is telling us is that portions of the immune system, at least, are downregulated under symbiosis. And that adds a new caveat onto our question of how is this cnidarian algal symbiosis established and maintained, but without sacrificing general organismal immunity? Because if this downregulation of NF-kappa B in the NF-kappa B pathway is occurring, how is that not a sacrifice to the overall organismal health? And so thank you to the previous presenters for giving a little bit of background on 10x single-cell RNA sequencing. I use 10x to uncover cell-specific differences in gene expression in symbiotic and aposymbiotic Oculina arbuscula in order to really determine these nuances behind the immune system under symbiosis. And so what you're looking at here is a uniform manifold approximation and projection, otherwise known as a UMAP. And in a UMAP, each of these dots here is the gene expression profile of one single cell. And this UMAP altogether is comprised of cells from aposymbiotic and symbiotic oculi neurobuscula. And the closer together cells are, the more transcriptomically similar they are. And so that's how we're able to cluster these cells by cell type. And we're able to really excitingly determine a transcriptomically distinct immune cell cluster. And so this is really exciting because it's one of the first instances of performing single cell RNA sequencing in a stony coral model. And we were super excited to find the immune cell cluster. However, when we looked at the transcriptomic profiles of the immune cells from our aposymbiotic and our symbiotic coral, they're not separating by symbiotic state, which was surprising to us because we see so much differences in immune system genes and proteins at the whole organismal level. So we went back to the drawing board and thought about these gastrodermal cells, because again, these gastrodermal cells are the cells that are hosting the algae. And when we look at the gene expression profiles of these cells, we see, probably not surprisingly, that they do separate by symbiotic state. And so we were interested to see what genes are driving this separation by symbiotic state. And so we broke down that UMAP into the principal components that are defining the UMAP. And so we broke that down further into the genes that are driving those principal components. And we see that a lot of the genes that are driving those principal components are involved in the immune system, and specifically might have roles in the NF-kappa B pathway. And these NF-kappa B response genes are downregulated in symbiotic gastrodermal cells in comparison to aposymbiotic gastrodermal cells. So this is the average log2 fold change on the x-axis. And when you have a negative value, that's higher expression than aposymbiotic compared to symbiotic. Furthermore, we looked at this is just a UMAP, again, of our aposymbiotic and symbiotic cells across the whole cell profile. And we took the cells here that have over 2% of their transcripts as corresponding to genes that have been previously annotated to be in the cnidarian immune system, co-expressed with NF-kappa B, and are higher in aposymbiotic at the whole organismal level. And we see that expression of these genes is higher in aposymbiotic gastrodermal cells. So overall, what this is telling us is some really exciting things. First, it's telling us that immune genes have distinct and cell-specific as well as symbiosis-specific roles, and that differential regulation of the immune genes in the gastrodermal cells may facilitate symbiosis and promote compartmentalized immune regulation. And so that's all I have for you, but thank you so much for listening, and thanks to all of our funding sources, as well as all of the labs that have supported us. Thank you so much.
Thank you, Maria. Questions from the audience?
That was a great talk. Thanks. How well does the 10x sequencing work on non-human organisms?
Yes. So this has been this has been such an adventure for us because it works. It works, obviously, but it takes a lot of effort, and a lot of the reagents need to be specifically characterized, or that's going to go off, for the coral itself, because coral is a marine organism, and so it has a different isotonic balance. And so when we work with the normal 10x reagents, and when we're isolating the cells have to be live when they're going into 10x processing. So we're isolating the cells. We have to be really, really careful about what buffers we're using. And so we have to be really careful about the salt content, because the coral cells need more salt than human cells, but then salt content messes up the 10x processing. So yeah, it works, but through a lot of effort. And we haven't had as much success with other methods like split-seq, which we've been trying to do with Aptasia, that sea anemone, which in split-seq excitingly involves fixation. But we've definitely struggled a lot more with that in our marine organisms. Yeah, so there's a lot of work and exciting processes to still be done.
Great. Thank you, Maria. And our final speaker for the lightning talk session is Jace Ruttenberg, also from the Department of Biology on the Charles River campus, talking about human viral transcriptional regulators. Let me get your talk up here. Sure. All right, there you go, Jace. Thank you.
Hi, everybody. Thanks for having me, and thank you for the introduction. So I'm really excited to come out here and actually talk to you guys about my project uncovering the functions of human viral transcriptional regulators. So just some background on this project. So viruses, they employ many different strategies in order to actually alter cellular environments and basically tip the scales in the favor so that they can actually properly grow and evade the immune system, things like that. One of the main ways that viruses do this is by deploying specific proteins that we dub viral transcriptional regulators, or VTRs, that can affect host gene expression and ultimately have effects on pathways downstream of that can lead to differences in disease outcomes. So VTRs, they affect expression through a few different mechanisms. They can act like host transcription factors and bind directly to DNA. They can also co-localize to regulatory elements with human TFs. And from there, they can enact on host genes and basically change different genes and different pathways depending on which virus wants to do what. So some previous members of the lab actually compiled a list of what we considered human VTRs, basically from different viral families, using both an extensive literature curation as well as homology-based analyses. And what we found is that basically almost every family of human infecting viruses actually code for VTRs, with some larger viruses, such as double-stranded DNA viruses, coding for many, relatively speaking, and then RNA viruses and retroviruses coding for relatively few, between 1 or 2. So, but we also noticed in our curation that there are a lot of holes here. There's really not a lot of information on the different types of genes that are targeted by VTRs in general across strains and species. We also don't really know what the mechanisms of action are for how they actually affect transcription. And furthermore, we don't really understand very well the transcriptional consequences on a broad scale for many VTRs. So one of the goals of our lab was actually to set out to figure out how this works and determine these things, and to answer the central question of, like, how do VTRs actually perturb host networks at large? So just looking at some of the data that already exists there, we actually see that there's very limited amounts of in vivo binding data for VTRs, really only representing 12 of them of the 400 some odd that we classified. Within this data, we can actually say that there's actually a pretty good number of variation in the amount of peaks that we see per VTR, ranging somewhere between 100 and 50,000, depending on the VTR, and that there's a pretty solid amount of variation in preference as to where VTRs actually like to bind, with some binding really close to the TSS promoters and some binding more distal elements, which suggests to us different mechanisms of action depending on the VTR. So in our lab, we employed basically a factor-wide approach rather than a genome-wide approach to try to assess binding of VTRs and how they bind human promoters. In this case, we looked at a subset of immune response and cell cycle regulation promoters, which are also enriched as targets in the existing ChIP-seq data. And what we found is that VTRs can either bind independently or in cooperation with human transcription factors. And this is using 2 different yeast-based assays to either look at single monomer binding or binding with a human transcription factor there. We could also leverage our paired data that I just mentioned to also see antagonism at human promoters. So this is basically what I'm showing is a relationship map looking at VTRs and human transcription factors, with blue lines representing cooperative binding events at a promoter and red blunt arrows representing antagonism. The thickness of these arrows basically showing you how many promoters this occurs at. We see a few interesting examples where basically you have a VTR. In this case, I'm looking at this EBNA3B and RBPJ axis here, where basically you can have a VTR that actually antagonizes binding at one promoter, but cooperates with that TF at another promoter. And basically what we think this is suggesting is rewiring. We think altogether this is like a small glimpse at like the underlying regulatory complexity just at the DNA level. So we see viral proteins messing with these networks and manipulating that, and also HTFs doing the same thing to them. A level up from that, we also used mammalian 1 hybrid systems to assess transcriptional activity of VTRs in human cells. We see that a lot of VTRs tend to activate transcription overall, but we do see some that also repress. And interestingly, when we break this down to orthologs of the same VTR coded by different strains of viruses, there's some inherent variation. So some actually have different effect sizes, depending on what the strain is, and some actually have different directionality of transcriptional regulation altogether. Looking at what might underlie this, we also went to protein-protein interaction data, both from the literature, and used a yeast-2 hybrid system to add to this network of VTRs and human proteins, and found that VTRs seem to like to bind or interact directly with human TFs, shown here in purple, and then also human cofactors, shown in green, which suggests to us, maybe unsurprisingly in some ways, but VTRs like to co-opt the machinery of their host. They like to use the same type of machinery that the host uses just to redirect and alter gene expression to their favor. But this left like another question for us as to what type of target genes are actually being dysregulated. To answer that, we use transcriptomics to basically assess the transcriptomic profiles of around 80 VTRs of the VTRs that we tested. And we see that a lot of the pathways, this is a heat map basically looking at up or down regulation of pathways rather than individual genes. And we see that a lot of the pathways that are related to viral replication and survival, such as cell signaling, immune and stress response, and even things like DNA damage repair and cell cycle, they're all regulated. They're all regulated a lot of times in, many of these pathways are regulated at the same time. And there are a few different modules we see where there seems to be some switching in what they actually do. This doesn't really cluster by viral family, which was interesting to us, or even virus. So we took a closer look. This is just like a different way of basically looking at this pathway analysis, where each point represents a different pathway. The coordinates represent the normalized enrichment score for that. VTRs from the same virus tend to have pretty different effects from each other. There's not really a correlation. Viruses tend to avoid functional redundancy, so that's kind of expected. Like we saw in our mammalian 1 hybrid data, you can have pairs of orthologs that are either quite different from each other in terms of the transcription response they elicit, or quite correlated. And surprisingly, when we look at some cases of VTRs from very different viruses, in this case, papillomavirus and a herpes virus, which are orders of magnitude different in size and infect different tissues, we can see that they elicit really similar transcriptional responses, which look as close to us as pairs of orthologs from closely related viruses, which is kind of suggesting that there might be some convergence at a sort of a strategic level of changing gene programs, rather than, like pairing with viruses. We see like a broader sort of change in the way that the transcriptomes are being altered in a virus to virus case. So altogether, I try to take some of this time to show you that VTRs, they can function by binding alone or cooperatively or antagonistically with human TFs, that they can interact with human TFs and cofactors, regulating transcriptional activity in a variety of ways, and that they can have various outcomes on gene expression, on genes that we expect, some that we don't. But I didn't have too much time to talk about it, but we also have a few different ongoing efforts to assess the impacts of different VTRs on things like chromatin state, and also to determine DNA-specific binding motifs that underlie TFs that directly bind. And so I just want to thank everybody in my lab, everyone who worked with me on this project, my collaborators, and you guys for taking the time to listen to me talk about this. Thanks so much.
Great. Thank you, Joyce. Questions?
Hi.
Hi.
Yeah, I have kind of a quantitative question, because I could imagine if you put enough of almost any random protein into a nucleus, it's going to affect transcription of a lot of genes. So what are the levels of some of these viral proteins in an infected cell, and how does that compare to what you might see with a kind of, typical specific transcription factor? And also, do you have any idea how the binding affinities compare?
Yeah, so that's a really good question. It's something that we're also very interested in. So in general, viral proteins, they have different expression levels, and that's typically correlated to what stage the virus is in, whether it's like latent and maintaining latency, or undergoing something like an active infection. In these assays, the expression is fairly high because we're basically just transiently transfecting with a human promoter. We also have several data sets I didn't really talk about where we have stably transduced cell lines that have varying levels of expression that we can control. In terms of comparing that to human TFs and proteins, it's something that we're trying to do. It's just kind of tricky because we're basically trying to pull out from the literature comparable data sets. But we do want to assess that because these effect sizes are really huge, and that's true. In terms of your question about affinities, so I mentioned briefly using a method to essentially look at the underlying DNA motifs. This is an in vitro method that was recently developed over in Martha Bulick's lab at Harvard Medical School. And one of the other features of this method that we really like is that we can also get binding affinities for these TFs to different oligonucleotide sequences. So we want to leverage both of those pieces of data together. I hope that answers your question.
Okay, thank you, Jace.
Thanks so much, guys.
And thank you to all the trainees who came and spoke today. I think the GSI judges will have our work cut out figuring out which one will win the top prize. Because please give another round of applause to our speakers. We're now going to transition to lunch and poster session and vendor show. At 2 o'clock, our dean, Dr. Karen Ammon, will present an address before the GSI invited faculty talks. Food is getting set up thanks to Checkers. Yeah, everything is good to go. Please get some food, visit our vendors, check out the posters, and again, check out this rotating.
It's okay.
And this will run in the background. Everybody, as we wrap up the poster session vendor show, I'd like to bring everyone back to the podium here in the screen. I'd like to introduce our Dean and Provost, Dr. Karen Antman, who will be addressing us. She is our biggest supporter of the Genome Science Institute, and she'll be addressing us as we then move on to the last hour of our 16th annual GSI Symposium. Thank you, Karen.
Thank you, Nelson. So it's a real pleasure to introduce you to the 16th annual Genome Science Institute Research Symposium, where every year we share your outstanding genetics and genomics research. It's hard to believe that we've been doing this for 16 years. Actually, this was one of the first centers that we got started. The problem was that we had genetics and genomics in multiple different departments, and therefore you really had to have a center. And unlike some of our other centers, which are only represented, the members are only representing one department or even one section, this one really is interdepartmental. One of the particularly nice and very civilized traditions in academia is enjoying lunch while browsing through 30 or so research posters by undergraduates, graduate students, postdocs, and faculty. So it really is one of those nice things that we do once or twice a year, looking over research. As you all know, I'm looking forward to turning over the deanship sometime in the spring and taking a real vacation. So someone else will be welcoming the symposia next year, but it's been a real pleasure supporting this center over the course of 17 years, because basically in the first year we didn't have a symposia. The first center started with Dr. Richard Myers. Do you remember him? He was the first director. And our goals then were to encourage new collaborations. Therefore, we needed a symposium, generate promising preliminary genetics data, thus a pilot grant program, and then to submit your great new research projects for NIH funding. I'd like to thank our current leaders, Dr. Nelson Lowe, Grishak, and Flynn, for taking on this commitment to promote both basic and applied genetics research, and also for organizing the symposium. It looks like it comes together, but it's actually a lot of work putting those things together. So to help you generate your labs generate preliminary data, the GSI provides workshop pilot grant competitions in the spring. So look forward to those in the spring for testing different genomics techniques here on campus. And you're certainly generating terabytes of data, as you've seen outside on the illustration. So we have research computing and also our data science core. So as the membership continues to grow at all the faculty levels, I look forward to seeing your outstanding genetics work, not only in the scientific literature, but also communicated to the lay press and into Bostonia and all the other BU publications. Please feel free to, and actually you should, contact Gina whenever you get a paper accepted. They will follow the embargoes and work with the journal so that you get your paper out to the biggest possible audience. And so the impact is bigger than if you don't take advantage of their services, because they will, and they'll do it all within the embargo. So you don't need to worry about that. That's the communications office. So finally, I'd like to thank our commercial vendors here who supported the symposium. We also actually depend on their research projects for the rest of the year, throughout the year. So thank you for coming to today's GSI Research Symposium. Dr. Lau?
Thank you, Karen. Okay. For the last hour here, we're going to have 4 talks by the newest members of the GSI. Our first speaker is Dr. Philip Muse from Department of Medicine, as well as Physiology and Biophysics. And he'll be talking about context-specific genetic and epigenetic regulation of patient heterogeneity in neuropsychiatric disorders. Let me switch over here. Get your talk going. This one, right? If you can
That was
use
a different title.
Oh, different title. Oh, okay.
That's
Simpler one. And here's the pointer.
Okay, thank you. Okay, really, I will be talking about epigenetic control of neurogene programs and drug addiction. Can you hear me okay? Is this on? No, this is fine. So, yeah, I'm really excited to be here and to be part of GSI. I've been really impressed with the research that's being pursued by everybody. It was really incredible science that I saw here at the poster session earlier at the lightning talks. And so it's particularly impressive to see this across so many fields. And so in my lab, our team is interested in the molecular basis of addiction. And so I'm going to start out with a pretty devastating number, especially as we know that there are millions of people who are trying to escape addiction every day. There are like 85% of people who come back home from rehab relapse. And so this really is a disorder, not so much where people battle willpower, but where our research has shown that they're deeply encoded biological changes that drive the motivation to seek out and to use drugs, even after prolonged abstinence. And so shown here is like one example of brain imaging showing brain activity in people with cocaine use disorder. And so what I'm going to talk about is some data that are pointing to like really persisting changes on the epigenetic level that allow for this encoding locking in these addictive behaviors. And I will discuss some of the approaches that we plan to take to unlock some of these changes again in new therapeutic approaches. And so what we view addiction as like generally a very complex disorder of learning and memory involving the mesolimbic dopamine system. And you see my mouse here. That's involved in pleasure and reward, that's governing goal-directed behaviors. And what is really striking is that these drug-associated memories and drug cues are incredibly powerful, and they can stay powerful throughout prolonged periods of withdrawal. So even after years of abstinence and years into recovery, they can trigger craving responses and relapse. And strikingly, this effect also incubates over withdrawal. So the behavioral and molecular responses we do see in relapsed patients or in our animal models are even greater. And so involving major changes on the level of gene regulation. And indeed, we knew for a long time that drugs of abuse trigger neuronal gene programs, the genes that are dysregulated, that's involving, dopamine triggered intracellular signaling cascades in these brain reward regions that then induce neuronal gene programs that encode proteins that sit at the synapse, receptors, transporters that lead to structural changes and synaptic plasticity. And our research and the research of others has shown that this transcriptional response is reliant on modifications of histone proteins. In particular, acetylation that we see greatly induced in the nucleus accumbens, which is receiving dopaminergic inputs, and it's like a key region of reward where histone acetylation opens up the chromatin structure and makes genes more accessible for them to then be rapidly transcribed. And so what's important is to know that the remodeling of the epigenome can have really long-lasting, persistent effects on gene regulation that outlive the initial transient signal of drug exposure. And so indeed, we saw this in our animal studies in mice that self-administered cocaine. These mice self-administered cocaine for 10 days, and then we put them through prolonged withdrawal. 30 days, they didn't see any drug. And we performed transcriptomics in the nucleus accumbens after giving them a dose of cocaine. And we saw a massive transcription response to that cocaine after withdrawal. And that was like greatly increased in comparison to drug-naive animals that have never received cocaine before, that, however, received the exact same dose of cocaine. And so we think that this priming response, that is like this dysregulation of gene programs, is driven by epigenetic changes that are part of the molecular memory, and that is established with repeated exposure to drugs and persist throughout withdrawal and underlies this like massive dysregulation that's linked to dysregulated behaviors and underlying craving and relapse. And so to get at this, we performed proteomics in the nucleus accumbens, looking at what proteins are actually recruited to chromatin to get some idea of like, okay, what's binding to genes in withdrawal, and then what genes are released into the nucleus soluble fraction. And we found that many genes involved in histone modification and epigenetic regulation are recruited to chromatin in the withdrawal state, involving not just histone modifiers, but also remodeling complexes like the switch-SNF complex, which some of you might know from your cancer studies, where you also see massive dysregulation of chromatin and dysfunction of gene programs. And then we performed more mass spec to look at all these different modifications, acetylation, methylation, phosphorylation. We described many changes on that level. But something that was rather unexpected with this like really unbiased approach that we were able to uncover is that certain histone variants were changed in the chromatin structure. And in particular, we found that cocaine withdrawal was linked to the depletion of H2AZ, which is a key histone variant of H2A. So I'm summarizing that finding here in this graph, where at baseline, we find actually H2AZ at key gene regulatory regions, like promoter sites, and that H2AZ binding is associated with lower gene accessibility. So in generally, these genes are not very accessible. Now in withdrawal, we find that H2AZ is kicked out of chromatin, out of the chromatin structure in the accumbens. And at that point, we also find increased binding of this very specific histone chaperone, M32E, that has been previously shown to be highly specific chaperone for H2AZ and is allowing its removal from chromatin. And so with that, at these sites that lose H2AZ, we also see increased accessibility. So those genes that lose H2AZ are more accessible. However, we don't see major transcriptional effects at that stage yet, in the withdrawal stage. Those become clear once we give a challenge injection. And then those sites, those genes that lost H2AZ, those seem to be primed for very speedy activation. So they're like ready to go and rapidly induced. They're accessible to transcription factors that are typically responding to cocaine exposure. And so these genes are involved in synaptic plasticity and many genes that we have previously linked to memory. And so it was really exciting to have this insight from our rodent models, but obviously we wanted to know whether those molecular changes also reflect disruptions that we see in people with cocaine use disorder. And so we were lucky to have access to brain tissue, nucleus accumbens from people with cocaine use disorder. And we also performed similar analyses on the brain tissue. We performed transcriptomics and found also genes involved in plasticity and memory to be dysregulated. And we did a big effort to do this interspecies transcriptional analysis to really drill down into what networks and gene programs are disrupted. And we indeed found that the molecular changes are highly conserved in our rodent models that we find in people with cocaine use disorder. What I didn't touch on yet is that these analyses also pointed to very specific subtypes of neurons that are involved. So the nucleus accumbens is largely made up of medium spine neurons that express either the D1 or D2 receptor. And we found that plasticity in the dopamine D1 receptor neurons is really driving these transcriptional changes. And in those neurons, we see this priming effect. And so we went back to our animal models and now blocked H2AZ removal in these dopamine responsive D1 neurons and knocked down M3A2E, that specific histone chaperone, and then looked whether that is altering cocaine self-administration in our mice. And so here in this graph, we show that when we make it harder for the mice to get cocaine, we put them on a different schedule. So they have to here FR2, fixed ratio schedule 2, press twice as often to get the same amount of cocaine. You see in the control mice, they elevate their lever pressing and they press more when we make it harder to get the cocaine. But we don't see that changing behavior in the knockdown animals. And so here we really show an example where we affect a key player in chromatin remodeling that is affecting behavior. And so I want to quickly discuss what we are doing now in the lab, building on this work and using new technologies, sequencing technology, single nuclear RNA-seq, and spatial transcriptomics to try to identify more specialized subpopulations of cells that really encode cocaine-related information that drive relapse. So we're looking at these cocaine ensembles, and using very specific transgenic mouse models, we can tag those cocaine ensembles using the ARC promoter that is driving D2 So the ARC gene is really interesting because that we found to be rapidly upregulated in all the neurons that respond to cocaine, that are activated by cocaine. So we're taking that promoter of that ARC promoter, now drive DD2 and make this system tamoxifen inducible. And now we are able to give cocaine and tamoxifen at the same time and label only the neurons that are activated by cocaine. And so this is a video that's not playing, but it looks pretty cool. So as you see flashing right now, this is calcium imaging from in vivo calcium imaging from our collaborator. And we can show that the activation of these very same cells drives relapse. And these investigations really revealed something very new and perhaps unexpected. It is that the refined ensemble that really encodes the memory the cocaine-related memory, and after repeated exposures to cocaine that's related to relapse, it's actually smaller. So there are fewer cells over time that more robustly drive behavior. And so we are doing now a lot of single nuclear RNA-seq to explore what's going on in those cells and what's going on in these other cells that don't respond anymore. So how they're shut down. And indeed, we already find key chromatin players that are recruited. And so the idea is that chromatin really acts as a platform for ensemble refinement and allows for these lasting changes. And ultimately, our goal is to repair these molecular scars, these chromatin scars, we dub them, to prevent relapse. And we are setting up these models right now. We're using CRISPR tools to edit the epigenome. And the idea is to reverse engineer some of these lasting changes we see in the cocaine ensembles to address drug-related plasticity and relapse, to recruit these specific effector domains to key genes to close them back up or do the opposite. And so with that, I want to give a big shout out to my collaborators here at BU across the States. And of course, my lab members, some of a terrific team, and I'm very happy to be here at BU and part of the institute. So thank you. Taking questions.
Thank you, Dr. Muse. Any questions from the audience? Oh, way in the back. Okay. Yeah.
Yeah, so this work was all done with cocaine. So the question was whether we see similar things with other drugs of abuse. My lab is using primarily cocaine and alcohol. We're funded by NIAAA. And so, yes, we're investigating also changes that are induced by alcohol. And in particular, we're very interested actually in metabolic pathways that support these epigenetic processes. So where do the acetyl methyl groups come from that we see changed in withdrawal? And we see really, truly these metabolic pathways as potential new drug targets that are easily druggable, because there's been a ton of work done in cancer. And so, yeah, that's kind of where we're headed right now. And we do see similar changes, at least in cocaine. Yeah, cocaine and alcohol, yeah. Yeah. Yeah, so cocaine is pretty much the cleanest and easiest drug to study. It's mode of action is very clear. Alcohol is pretty messy, but looking at, we're just looking at one particular subtype here in the nucleus accumbens, we do detect similar changes, but obviously that looks a lot more complicated as we move to other brain regions. Yeah.
Well, two more questions over there. And over
Philip, great talk. The HLA-Z change that you see would seem to me to cause a generalized dysregulation.
Yeah.
But I associate changes in addiction with also more kind of neurophysiological specific. So how do you reconcile those two?
Yeah, it's really, really bird's eye view, and it's a lot more complicated, especially with H2AZ, where we see it's actually not just that wherever H2AZ sits, genes are not accessible. Its acetylation status itself is very relevant, and that's highly regulated. And so, yeah, what was surprising to us was that we saw this general opening of chromatin genome-wide, even in genes that are generally responding immediately to cocaine in drug-naive animals. And it's interesting, I don't know how exactly that is happening, but similar things have been reported in hippocampal cells in the context of fear conditioning, where you see genome-wide opening and accessibility increase. And they're now genes primed, so to say, to that are more accessible to transcription factors that would never actually go there in the first place. And again, like when and how this happens, and whether we're looking always at like one particular point in time, like 1 hour after we give the cocaine, perhaps we just shift the whole system in time, like at earlier or later time points. It's not so clear. So how this exactly happens, and like what's recruiting these remodelers to those genes, and like it's not very clear. But yeah.
One more quick question over
Yeah.
here.
In your mice, have you seen evidence of transgenerational epigenetic inheritance of cocaine addiction?
Yeah, no, we're not. That's super interesting, but we haven't looked at that. Yeah, that's really, yeah, and obviously this points to an effect like these lasting changes on the level of chromatin. Early life stress, they might be influencing gene responses later in life. And so if you view chromatin as an integration platform for all these signals coming in and like insults that you might accumulate over lifetime during like important windows, they will have an effect much later. So that's the take home. So, but like we didn't look transgenerationally. Yeah, thank you.
Thank you, Dr. Muse. Okay, our next speaker is Dr. Stefan Isaac from Department of Biochemistry and Cell Biology. And he will be telling us about the mitochondria. All right, there you go. You can use this too to pointer.
Awesome. All right, hopefully everyone can hear me. Wave at me if you can't. Thank you very much for coming. Today I'm going to talk to you about how we can use some of the latest single molecule and like so-called third generation sequencing technologies to start to understand some complex genome biology and even protein mechanism. And the genome that my lab is particularly interested in is that of the human mitochondria. So you might be aware that we have mitochondrial DNA, but maybe you don't think about it too much. This is an image of a mammalian cell. And I just want to point out that in blue over here, that's the nucleus where all of the chromosomes that you typically think of are. Now, the mitochondria are labeled in red. They form this really vast dynamic network that extends throughout the cell. And all of those green dots that you see distributed throughout that network are copies of mitochondrial DNA. So whereas in the nucleus, you typically have 1 or 2 copies of each chromosome, a typical human cell has on the order of thousands of mitochondrial DNA. And so what does this genome do? Mitochondria have a lot of functions, and one of them is that they produce energy in the form of ATP. This is done through a series of chemical reactions carried out by called oxidative phosphorylation, carried out by these so-called OXPHOS complexes. It's 5 protein complexes that sit in the inner mitochondrial membrane. Now, these complexes from a gene expression point of view are incredibly interesting because they are encoded by 2 physically separated and evolutionarily separated genomes. So we have about 100 proteins here. Most of them are encoded in the nucleus. So these go through your standard, they're transcribed by RNA Pol II, they're spliced, they're exported, they're translated, and then those proteins are imported. The remaining, and in humans, that's 13. That's what that number is under this thing that won't go away. There are 13 subunits encoded in the mitochondrial genome. And so these are transcribed by a dedicated RNA polymerase. They're processed. They are translated by unique mitochondrial ribosomes and embedded in these membranes. And so this genome, its whole job is to make these 13 proteins, but they're all absolutely essential for oxidative life as we know it. And so 13 genes might not seem like a lot, especially when you think about the fact that, we have somewhere around 20,000 genes or something like that. But in highly energetic cell types, as much as 30% of the cellular mRNA can arise from mitochondrial transcription. So this is a very active genome. So what does this genome look like? Here's a little cartoon schematic of it. As you can see, it is circular. It arose from a prokaryotic endosymbiotic event over 1 billion years ago. And it's very small. It's only 16.5 kilobases. It's basically a plasmid. It's highly compact. So pretty much the whole thing encodes something. We have 2 ribosomal RNAs, 22 tRNAs, 11 mRNAs that encode 13 proteins. And then we have 1 non-coding region up here in gray. And this is our control site where we have our transcription start sites, our origin of replication. And so really that's where the activity of this genome is controlled. And then, of course, one facet of this genome that I'm fascinated by is the fact that it is highly polyploid. So you have thousands of copies of this genome in a single cell, which really begs the question of why and what are all of these genomes doing? And so one aspect of biology that I'm really interested in is actually the physical packaging of DNA and how that is used to control genome activity. This has been achieved, if you look across life, different ways. So if you look in the nucleus, DNA is wrapped around histone proteins. These form nucleosomes, that forms chromatin fibers. Bacterial genomes are organized into nucleoids by nucleoid-associated proteins, but mitochondria exist in this weird space between the two. They arose from a prokaryotic ancestor. They've evolved in the context of a eukaryotic cell, and they actually use machinery that's distinct from both of them. So you will not find histones in mitochondria. You will also not find bacterial nucleoid-associated proteins. They have all of their own stuff. And so really, when I started out, the question I wanted to get at is actually how is this genome physically packaged in vivo? Because I realized that we didn't actually know that much about this, even though despite its size, I figured we would. And one way that you can assess this is by measuring DNA accessibility. So the quick version of this is that if DNA is inaccessible because it's bound by a protein or what have you, then some other factor is not able to bind and perform its function. And if DNA is accessible, then this blue protein is able to bind and act on the DNA. Whether or not we're talking about a polymerase or a nuclease or a helicase, they actually need to be able to physically get on the DNA to act. And so this gives you a sense of the regulatory capacity of a genome, whether or not a gene could be on. Now, there have been a lot of techniques to study this that have been developed, which have been super informative for primarily the nucleus. And you've probably heard of some of these. There are things like DNA-seq, MNA-seq, ATAC-seq. They all work by the same principle, which is that you have some sort of enzyme that is going to cut the DNA where it's accessible. That's the green Pac-Man. You get a bunch of fragments. You then use short read sequencing, and then you can map the ends of those DNA. And you get this sort of averaged accessibility map in this cartoon that I've drawn, where you can see where the genome is more or less accessible. These are great. You don't need a lot of material. They work genome-wide. But the problem is that for some systems, that it's a bulk profiling method. So any sort of significant heterogeneity is going to be obscured. And when we're looking at mitochondrial DNA, if we need 100,000 plus cells, maybe millions of cells, and we have 1,000 copies in every cell, then we're going to potentially have a lot of heterogeneity. And so this doesn't work in mitochondria, which I'll show you in a minute. But basically, to cut to the chase, we needed to develop a new technique to do this. And we wanted to be able to measure the DNA accessibility of single molecules. So what we developed is this approach called FiberSeq. And rather than cutting the DNA with a nuclease, we're going to modify the DNA. So here we have a non-specific methyltransferase. And what this enzyme will do is where it is able to access the DNA, it will catalyze adenine into N6-methyladenosine, or M6A. That's the little purple stars. We can then isolate these high molecular weight DNAs. They retain that covalent modification. And then we can sequence them. Now, everything I'm going to show you today was sequenced on the PacBio long-read sequencing platform. But I want to also say that this is possible on the Oxford Nanopore platform, and we are going to be setting that up in the lab soon as well. And so these technologies allow us to get reads that are very long, greater than 15 kilobases, a lot greater than that. And we can identify where these methyl groups are. And so we can see where the DNA was accessible. And so now I'm just going to show you what some of those data look like. So I'm visualizing the genome linearly, even though it is a circle. So it goes back around to the other side. So the top green track, that's ATAC-seq data. And so you can see by ATAC-seq, the genome looks more or less equally accessible everywhere. Now, if we look at our methylations, they kind of reflect the same thing if we pile those up. But we actually have single molecule resolution that we're able to achieve here. So what I'm showing you here are just 80 randomly sampled reads. And for a typical sample, we have somewhere between like 50,000 and 200,000 reads. And so each horizontal line here is an individual read. So that's a single genome that we purified out of the cell, extending across the entire 16.5 kb. Now, all the little purple vertical hash marks that you see, those are where we identified those methyl groups, which tell us where the DNA was accessible. So what we were able to achieve here is both single genome resolution of DNA accessibility at nucleotide resolution. Now, one thing that you can probably take away from this is that there's not a lot of purple. And so most of these genomes have very, very little methylation, indicating that they are just inaccessible. And we can quantify that. And this is just a histogram from 6 biological replicates. I should say that this is from HeLa cells, and I'll come back to that in a second. So what we see is that in these cells, about 80% of these nucleoids have background levels of methylation being detected. So most of these genomes are just in a completely inaccessible state. And by first principles, they're probably completely inactive. The remaining genomes, and this is actually just a really long distribution, have varying degrees of methylation and accessibility. And so you might be wondering, is this just a weird phenomenon about HeLa cells? But we've done this in a number of cell types. We've done this in cancerous cells, immortalized non-cancerous lines, primary lines. And in all situations, it seems like this phenomenon holds that the majority of nucleoids are in this inactive state. Now, there's a lot of information here besides just looking at how much methylation is on a genome, and we'd really like to tap into that. But that's hard to do computationally. We have a lot of reads, and by eye and manually, that would be very challenging. So what we did was we developed a machine learning-based approach using a hidden Markov model that essentially converts these methylations into this footprint space that allows us to see where genomes were protected. So here I'm showing you on the right 250,000 reads. This is a heat map, where purple shows you where the DNA was accessible, orange to gray is inaccessible, where the gray means 500 bases or larger. So you can see out of these 250,000 reads, most of them are just blocks of gray because as you saw from this purple methylation, they're inaccessible. And so just because it's more interesting, we can zoom in on this population down at the bottom that actually has something going on. And so what I'm showing you here are 12,000 reads. I don't know how well the color is showing up there. And so these are reads that have greater than 10% of their adenines methylated. And so now we have essentially a footprint map of the entire bound proteome on the mitochondrial genome of all of these genomes that we isolated from cells. And we're able to dive into this and understand more about what's going on. And in the interest of time, I can't go too much into it, but I'll just point out 2 sites that are particularly interesting. One is right here at position 0 in the genome. This is a structure called the D loop. This is a product of replication, initiation, and pausing. And so we're able to look at changes that are happening here due to the DNA polymerase. And another site is over here at about 3 kb. And this is the binding site of this protein known as mTORF1, which is a transcription termination factor. And so this was really a sanity check that we could see that this known protein is binding here. And we actually see that it's bound in most of the reads, which is something that's possible with this single molecule technology. Now, I don't have too much time to go into all of the different data that we're able to. That's just sort of the baseline situation, but we're able to take this technology and use it to study mitochondrial gene expression more broadly, which is something that we've already published on, and now we're continuing to do in the lab and explore more systems. And some of these are oxidative phosphorylation dysfunction. So what happens when these systems break down? How do you change gene expression when you need to upregulate or downregulate how much OXPHOS is going on? What about just due to cellular energetics? So one thing that is in the published paper you can go read about is when you have these human skeletal muscle myoblasts as they differentiate into these multinucleated myotube cells and become much more reliant on mitochondrial activity. And then also studying more about, we don't have histones, I told you earlier. What mitochondria have is this factor known as TFAM. And so understanding how this protein functions to both package and regulate the genome using both cell culture systems as well as in vitro, like protein biochemistry in vitro in a test tube, to understand how this protein functions. And I didn't have time to show it today, but we have a really cool system set up where we can do this assay on in vitro reconstituted nucleoids, where we combine mitochondrial DNA with TFAM or any proteins of our choice. And so I'm just going to briefly summarize. What we're able to do is we can map mitochondrial DNA accessibility at the resolution of single genomes and at a single nucleotide resolution. We see that most of these genomes are inaccessible and inactive in human cells. And the ones that are accessible do seem to have hallmarks of both active transcription and replication based on those footprinting sites. And just to acknowledge a few people, so my lab is very new. I want to give a shout out to my research technician, Maël, And I have currently one rotation student in the lab, Sailor. I also want to thank Sterling Churchman, my postdoctoral advisor, and a number of people in the lab. Particularly want to give a shout out to Tommy Tullius, who was a bioinformatics graduate student in the lab, who really developed this machine learning hidden Markov model based approach, and our collaborator, Andrew Sturgachis, who is using FiberSeq to study nuclear structure. And I'm happy to answer any questions. Take my slides.
Thank you, Dr. Isaac. Questions?
Very nice talk. So I have two questions. One is that for each mitochondria, how many genome they have? Is it one or multiple?
So the question is, for each mitochondria, how many genomes do they have? So because they are constantly moving and fusing together and dividing apart, it's really rare that you'll have a circumstance of like an individual mitochondrion. But when that does happen, they do typically have multiple copies. If they don't, they can either be destroyed or reintegrated into the network.
And I guess another question is that when you see some mitochondria is really inaccessible whole genome-wide, so does it mean the whole mitochondria is inactive or doing something also in fusion, busy with fusion or fission?
Yeah, so you're saying if the genome is inactive, is the whole mitochondria inactive? No, probably not. I mean, because this is just like the output of transcription and replication as needed, but the mRNAs have some sort of half-life, the proteins have some sort of half-life. So you still have all of those proteins in mRNA that can be translated and functioning, even if the genome isn't on. And so what probably is happening is they're turning on throughout the network as they're needed.
I was just reading this morning about how mitochondria within a cell have different functions. Do you see evidence of that in your sequencing?
So by this, it would be hard to capture. So the question is mitochondria having different functions within the cell, and do we see that? This, because we are just grinding up our cells and looking at all of that DNA, we're losing any sort of spatial resolution, which is where that's going to come in. So it'd be super cool to look into that, to divide up subpopulations of mitochondria, because they're all over the cell, they're touching different organelles. We're not able to see that in these data, unfortunately, but that'd be super cool.
So cells growing in kind of standard culture media don't actually have particularly active mitochondria. They turn off oxidative phosphorylation.
Right.
Were your HeLa cells actively running oxidative respiration, or were they just kind of growing in normal media?
These are growing in normal media, like your standard DMEM. They have OXPHOS, they have those subunits. They are using that. They still use it, but you know, they're a little more amenable to that not being so great because they can do glycolysis.
Yeah.
So we do have a system set up where, you know, you can shift them into things that force them to rely on OXPHOS, like shoving them into galactose media instead. And yeah, we're exploring that more now. So we do have some preliminary data of like giving them only galactose, or, you know, be interesting to starve them too and see what happens.
Great. Thank you, Dr. Isaac. Third speaker is Dr. Li Hu from Biomedical Genetics. I apologize to Dr. Muse. I did get his talk title wrong because we had some, you know, but you guys, so your talk actually was the title I said mistakenly, but you'll be talking about patient heterogeneity. Yeah, let's see if this will work. Oh, shoot. No, sorry. It's stuck. I'll bring that up to EduMedia next time.
Thank you for this wonderful opportunity. I'm Lei. And coming from medical genetics section, our section is mostly working on the Alzheimer's disease genetics, but also other brain-related disorder. And I think it's very understandable we confusing the title with me and Dr. Mewis, because we are all working on epigenomics. And actually, we have collaborations. And my title is context-specific epigenetic regulation, and also try to see the implication in patient heterogeneity.
Use a microphone. Yeah.
Okay. And I go first with my acknowledgement. So previously, I'm working on at Manuel Kelly's lab at MIT. He's mostly working on epigenomics, single-cell omics. And then I extend a little bit to understand the genetics of histone modification. So that's the EGTEX, Enhancing GTEX project, and collaboration with MIT and Broad. and also try to understand more about the histones and the genetics, and in the context of immune cells in bipolar disorder. That's a collaboration with Mayo Clinics. And then we also want to go further about the high resolution, go to single nuclear RNA-seq to understand the eQTL in so many samples, brain samples with Li Hui's test lab. And that's my starting my new lab. And we have a bunch of postdoc and master students working with me and working on the genetics, epigenomics in specific contexts. And also we have privilege to working with Jenna and with Ben here and collaborate with M6A to understand whether there's a crosstalk between epigenomics, alternative splicing, and M6A. And I got a fully support with my section chair in the Federal. And yeah, so we're here very interested in complex disease, which means it's not about a single factor, but a lot of genetic loci are taking higher risk on the specific disease. And beyond that, also a lot of epigenomic factor, environmental factors there. So putting together, they are pushing the disease pathogenesis into different aspects. And one of the most important ways to understand that is through genome-wide association studies, through which people try to IC the genotype across different individuals. Now usually like millions of people, tens of thousands of people to millions of people for certain disease, and we check the association at the genetic variants, loci by loci, with the specific disease. And the consequence is that we identify a lot of GWAS loci there, but a lot of them are sitting at the non-coding region. So that raises a lot of challenges and opportunities. For example, what are the causal variants? Because a lot of genetic variants are co-inherited. And another is that even we know the certain genetic variants are associated with disease, so what are the specific cell types are those genetic variants are working at? So people usually using epigenomic features which shows a lot of cell type specificity, and try to using enrichment, try to see which cell types are enriching certain kind of genetic variants. And the third one is the one I'm most interested in, is about what are the mechanisms, which means that you know the genetic variants, and then what are the causal genes the genetic variants try to regulate at, and then lead to the phenotype. And usually people using eQTL mapping, which means they collect a bunch of sample across individuals at certain tissues, and then you mapping the genetic variants are associated with gene expression. So if the specific genetic variants are associated with gene expression, then it's called the eQTL. And that if that signal is aligned with the GWAS signal, which means that specific genes are likely to be the causal gene. So that's a very simple way to understand or to dissect the disease-associated genetic variants. And specifically, there was a lot of big studies working on eQTL, for example, genotype tissue expression project that cover a lot of, discover a lot of QTLs across different tissues. But there's also challenge and the limitation there. People find that the GWAS signal are not always explained by eQTL. So there are certain barriers there, and maybe there's something missing, and people think about there could be some contexts are missing. That's why the eQTL are not discovered at those healthy baseline tissues are may still there in certain contexts. And that's why we try to take approach to epigenomic approach, and leveraging the epigenomic variation both across the different tissues and across the individuals, and to understand both context and mechanism at the same time. And we developed the framework for 3 steps. And the first step is try to identify which enhancers are active through different tissues. For example, here we identify using A3K tensor isolation chip-seq data, which is a primary mark for enhancers, and to identify more than 200,000 active regulatory elements on the genome across 4 tissues. And then we also leverage reference epigenome to annotate those kind of ARE activities. And we found we identified 127 modules, and coming from 13 groups of those AREs. And we can clearly say there are certain groups are super active across different conditions, and there are also groups active in multiple conditions. And there is also a condition or context-specific group. For example, these AREs are only mainly active in immune system, and those are mainly active in lung, and some are active mainly in heart and muscle, some are mainly in brain. So this is a way we give the annotation and to tell apart which genome are more likely to active in what kind of condition. And that also provide a context for the specific genetic variants of overlapping with those kind of regions. And next, we also, based on that kind of information, we could check the enrichment. And one of the interesting findings is that schizophrenia and bipolar disorder, which are two psychiatric disorders, and their GWAS signal are actually enriched in the blood and immune cell types. So that's mainly coming from brain, those ARE detected in brain, but primarily active in immune cell types. So that gave a hypothesis that maybe in schizophrenia and bipolar disorder, immune functions are quite important. And later, we also tried to mapping the QTL for the histone modification, and we do find certain QTL are shared across different tissues, and some are very specific in for the specific tissue. And that gave another idea of where the tissues, those genetic variants can function and act. And specifically, we checked the 54 schizophrenia GWAS tissue and GWAS variants that are co-localized with our histone modification signal. And we found there are certain variants, for example, sitting in the core7 locus, they are specifically active as a histone mark in brain, and EQTL also in brain, but do not have any signal here in any other three tissues. So that provides context-specific regulation there. And also, we try to leverage the genetic signal, not only histone modification QTL, but also EQTL together to build our own pipeline to building the connection between cis-regular element to the gene. And then we also benchmark to other gene enhancer linking pipeline and found that we are able to discover more genetic signal. And that's the first part. And then we try to follow up with the enrichment hypothesis about schizophrenia and bipolar disorder immune enrichment in those kind of two diseases, and try to say what is the best way to further understand what's the underlying mechanism. And then since it's really related to immune function, so we designed the project really based on the circulating immune cells. And we ask whether there are certain roles there in bipolar disorder. And we collect those blood samples from the cohort from Mayo Clinic with both bipolar disorder and control samples. And here we chart 4 major histone modification and with ChIP-seq data. And together integrated with other informations. Again, we're using a similar strategy. We check what are the enhancers detected in those samples and whether they are active in other tissues. We also found there was a strong enrichment of the immune specific one. We also found there was something are detected in the blood, but primarily active in brain tissue. And consistently, we found that there was an enrichment of those ARE that detect in blood, but primarily active in brain tissues, also enrich the bipolar disorder signal and the schizophrenia signal. So that indicate there were some connections between brain and blood. And we also follow the same strategy for histone QTL analysis, and we found there were some genetic variants seem to impact multiple histone modification together, and they all convert to the endoplasmic reticulum function. And we also follow the same strategy to link the genetic variants and cis-regular element to the gene with a genetic evidence. And we are able to identify the downstream target and causal genes. And we found that there are certain genes, they are all supported by the QTL evidence from the brain data, which means those genes are more likely to be shared between blood and brain. We also found certain genes are more immune specific, and it was only discovered from the blood-specific EQL data. And further, we try to see, could we leverage those epigenome variation to understand bipolar disorder patient heterogeneity? We're using latent factor analysis and to identify certain factors there, and then grouping the patients and control samples all together, and finally form 5 different groups with very different proportion of bipolar disorder in each group. And then we integrate the data with the patient group, with the patient EHR data, and we found that 5 groups are enriched for very different terms. Group 1 and Group 2 are enriched more infection, inflammation-related EHR terms, and Group 3 are related to glucose intolerance related to diabetes, and group 4 is more enriched for quetiapine, which is a part of which is a routine bipolar disorder drugs and hypertension. Group 5 we do not see very strong enrichment, and those ones we also follow that up with like put the 5 groups into 2. One is infection related, another is infection non-infection related. And when we also discover drugs based on the differential epigenomic signatures, and we found there are certain drugs are previously, for example, targeted to the cancer cells, cancer or cardiovascular system. They are also suggested as a bipolar disorder drugs. And of course, we also identify the drugs for the bipolar disorder previously. So which means that we leveraging the data in the blood, we can suggest certain drugs that are potentially impactful for the bipolar disorder therapeutics. Yep, and a brief summary. I hope my presentation gave you a whole picture of what's going on in my group. And mostly we're interested in the system level precision medicine and try to understand the disease-driving context-specific molecular networks through the integration of genetics and epigenomics, and then try to build in a connection, for example, between brain and blood to identify the biomarkers. And we also like to validate our results and with our collaborators. And next, what we try to do is that, so histone modification is working, is acid at the bulk level, and then we are trying to looking at more single cell level. So that's why we're starting to mapping EQTL at the cell type level across the different cellular contexts and health conditions, environmental exposures. And for the we also want to understand the logic of the EQTL, how does it function, why it is so dynamic. So that's why we try to build the transregulatory network. That's the work led by Ting, my postdoc. And then we try to see epigenomically what are the transfer factors are binding at the different loci across different contexts, for example, inflammation, maybe later addiction, and then try to understand how the EQTL are sometimes targeted to one gene and sometimes targeted to the other gene for the same loci. And the third one is that we try to understand beyond epigenomics. So is there any crosstalk between M6A, RNA modification, and alternative splicing? We leverage the data generated in Wallenzin's lab and try to see that they are mostly working on exon signal, but we'll try to say, what about the intronic M6A signal? Is it related to alternative splicing? The cardiac DARC signal can give us provide the opportunity to simultaneously understand, quantify M6A and alternative splicing at the same time, and later we try to cross-reference with other epigenome data in Alzheimer's disease and try to understand whether there are some crosstalk there. Yeah, and that's it. And thanks for your attention.
Thank you, Dr. Ho. Questions in the audience? I'll start with one. So how many genes are actually conserved in or preserving the epigenetic state that you see in the bipolar disorder brain versus the blood? Are those genes that are commonly expressed between blood and brain, or are they like, how is that getting preserved? That's pretty amazing that that's the case.
Yeah, I think this is something we're quite excited about. So for here, it's like around maybe 10 genes. And so this is the gene not only expressed in brain and blood, but also the EQTL, which means the genetic variants are associated with those genes, both in blood and brain. And interestingly, those genes are mainly from the excited neuron layer level 2 to level 4. Layer 2 to layer 4, which is presumably the major source of bipolar disorder where it's impacted. So this is something that we also think about why there are certain genes that are connected between blood. They have shared a similar regulatory mechanism. One hypothesis is that the specific regulation, for example, ER, stress, ER function, ER homeostasis, maybe it's shared between immune system and the neuron system. That's why there is something shared. Yeah. So this is something why we like to go further and to see what's going on. But we also see there is something that quite immune specific. So which could mean that the function are more likely coming from circulating immune cells, and those kind of immune function finally put a stressy input.
And what are those structural variants? Are they small SNPs or are they big deletions?
Yeah, so
those?
currently what we discover is more SNPs. We haven't for the structure variants, but this is something quite important and interesting to follow up.
Okay.
Thank you.
No question?
Is this a mixture of both type 1 and type 2 bipolar? Have you broken it down
Next
to look
question, sorry, it's only type 1.
It's only type 1, okay.
Yeah, thank you.
Okay. If there are no other questions, let's thank Dr. Hu. And last but not least, we have Dr. Thomas Clark from Department of Pathology and Lab Medicine. I apologize, we have some like mix-ups on the brochure, so in terms of affiliations, but we're excited to hear this last talk by Dr. Clark about identifying novel chromatin-associated proteins in DNA damage repair. Let's see, this laser pointer, I don't think we can get rid of that pesky thing. So, sorry.
Thank you very much to the organizers for the invitation and for putting together this really fantastic symposium today. Yeah, I've been very impressed by the breadth of science going on within the Institute. So it's great to be a new member of this Institute. So today I'm going to talk a little bit about a new chromatin-associated protein that we identified that we think is important for DNA damage repair, and potentially it has implications for a human developmental syndrome. So my lab is particularly interested in studying how DNA damage is repaired. And just as a brief introduction, why we think that's really important is because unrepaired DNA damage can result in gross chromosomal aberrations and mutagenesis, which can result in malignant transformation and the development of cancer. But it can also lead to cellular senescence and cell death, which can lead to premature aging and prodroid syndromes, developmental syndromes, as well as neurodegenerative disease. So understanding how DNA damage is repaired can help us understand the fundamental etiology of several different types of human disease. So DNA is wrapped around the histone octamer to form the nucleosome, which constitutes the repeat unit of chromatin. Chromatin is then looped and packaged into chromosomes, and this is where obviously our genetic information is stored in the nucleus of cells. And in recent years, chromatin structure and function has been shown to be absolutely essential for DNA damage repair and for also maintaining genomic integrity. However, trying to identify novel chromatin-associated proteins using traditional siRNA or CRISPR knockout approaches can be quite challenging because many of these chromatin-associated proteins and chromatin factors are essential for cell viability. So they actually drop out of the screens before we can assess their function in DNA repair. So to try and circumvent this, what I did when I was a postdoc over at Mass General is we developed this Chromorphium library. And what this is a human open reading frame library of around 340 different factors. So within this library, we have known histone modifying proteins, known DNA binding proteins, as well as many non-annotated proteins that we would predict interact with either DNA or chromatin based on domains within their sequence structure. And these include things like PhD domains, zinc finger domains, and Tudor domains. So this is a cDNA expression library, and importantly, each construct in our library contains a V5 tag sequence. And that's really important because it enables high throughput immunofluorescent staining with a single antibody that recognizes that V5 tag. And that's really important in the context of these non-annotated proteins where the reagents available to study them are quite poor. So we combine our Chromorphium library with this high throughput DNA damage screening approach. So in a 384 well format, we have U2OS cells, and each well of our 384 well plate expresses a different one of our constructs from our library. So after a period of antibiotic selection to make sure that our constructs are expressing, we then pass a UV laser through each well of the 384 well plate to generate subnuclear microirradiation. And then after a short period of time, we fix the cells and we perform immunofluorescent staining. So we stain for the DNA damage marker, gamma H2AX, and that essentially marks the path of our laser. So we know where the laser has passed through and damaged the DNA. And we also stain for V5, which detects our construct of interest. And a positive hit on our screen is when we see co-localization with the V5 tag and our gamma H2AX signal. And that means that our construct of interest has been recruited to the site of DNA damage. And when we did this, we identified several novel chromatin-associated proteins that were recruited to sites of DNA damage, including this gene of unknown function called zinc finger 280A. And that's what I'm going to spend the remainder of my time talking about today. So the first thing we wanted to do was confirm the results of our screen, showing that ZNF288 was indeed recruited to sites of DNA damage. So we can see here a V5 tagged ZNF288 construct recruited to this laser-induced DNA damage as marked by gamma H2AX. And we then wanted to confirm this using several complementary approaches. So here what we've used is a cell line where we can engineer a site-specific DNA double-strand break, which is visualized by this 53 BP1 foci here. And we can see that an antibody that detects endogenous ZNF288 is co-localized with this 53 BP1 signal, indicating it's recruited to that DNA double-strand break site. We then irradiated cells and isolated the chromatin fractions at different time points. And we could see an enrichment here of ZNF288 about 45 minutes following ionizing radiation. And it seemed like quite a dynamic recruitment to chromatin and removal from chromatin. And we subsequently confirmed this with live cell imaging. So we see a very dynamic recruitment of ZNF288 to DNA damage sites using a GFP construct as well. So we know ZNF288 is recruited to sites of DNA damage. Is it actually important for DNA repair? So to begin to look at this, we performed classic clonogenic survival assays in response to ionizing radiation and a topoisomerase inhibitor called etoposide. And both of these agents are known to cause DNA double-strand breaks. And what we can see is that cells depleted of zinc finger 288 are hypersensitive to these agents. Now, to look at this in more detail, what we then did is we performed a time course analysis in response to ionizing radiation of the double-strand break marker 53 BP1. And what you can see here in the red bar, which is our ZNF288 depleted cells, these 53 BP1 foci persist, indicating the failure to repair these DNA double-strand breaks. And if we take a closer look at this in metaphase spreads and we actually visualize the chromosomes themselves, we can actually see a large increase in the number of chromosomal aberrations in the absence of ZNF288. So we really think ZNF288 is important for repairing these DNA double-strand breaks. Now, DNA double-strand breaks are some of the most deleterious types of lesions that a cell can experience. And because of that, cells have developed two major pathways to repair this type of DNA damage. So we have non-homologous end joining or homologous recombination. And homologous recombination is limited to late S and G2 phase of the cell cycle, where it utilizes a sister chromatid as a homologous template. And that's generally considered to be a more error-free repair process as a result. Now, because of the recruitment kinetics we see of ZNF288, we reason it's more likely involved in this homologous recombination as opposed to the more rapidly utilized non-homologous end joining, which typically happens within the first few seconds or within a minute of the double-strand break occurring. So to start to look at this, what we did is we looked at the recruitment of the critical homologous recombinase RAD51 in our ZNF288 depleted cells. And we can see quite clearly that in the absence of ZNF288, there's a reduction in these RAD51 foci. And this is completely rescued when we reconstitute with our wild-type zinc finger 288. We also then looked and used a quantitative method to assess the efficiency of homologous recombination. And we did this using a GFP reporter assay. And we saw using this assay that there was about a 70% reduction in homologous recombination efficiency in the absence of ZNF288. And we also validated this by looking at sensitivity to a drug called olaparib, which is a PARP inhibitor. And it's known that cells which are defective in homologous recombination are exquisitely sensitive to PARP inhibitors. And that's exactly what we see here with our ZNF288 depleted cells as well. So we really think that ZNF288 is really critical for this homologous recombination process. So a major prerequisite for homologous recombination is the generation of single-stranded DNA through a process called DNA end resection. And what that does is it creates these single-stranded DNA overhangs, which are then bound and stabilized by this protein complex called RPA. RPA then acts as a platform for the recruitment of that critical recombinase RAD51. So we wondered whether the absence or the reduction in RAD51 we see could be because of a problem with this DNA end resection process. So what we did is we looked at the recruitment of that RPA complex after ionizing radiation, and we could see that that was dramatically reduced in the absence of ZNF288. And again, we could completely rescue this by reconstituting with wild-type zinc finger 288. And if we look in the chromatin fractions, we can also see less total RPA in the chromatin fraction and also less phosphorylation of RPA in this chromatin fraction as well. So we think that ZNF288 depleted cells are defective in this DNA end resection process. So to try and mechanistically understand what might be happening here, we performed these electromobility shift assays to look at the binding of recombinant zinc finger 288 protein and either single-stranded or double-stranded DNA. And to our surprise, we were able to see that actually ZNF288 has a much higher binding efficiency to single-stranded DNA as opposed to double-stranded DNA, which was actually the complete opposite result of what we were expecting, because we think ZNF288 is actually important for generating this single-stranded DNA through end resection. So this is when we had to kind of scratch our heads a little bit and try and figure out what might be happening here. And we recognize that DNA end resection happens in 2 stages. So we get resection initiation, which is primarily mediated by MRE11, which is an endonuclease. So MRE11 creates a short section of single-stranded DNA. And based on this assay, we think that is the substrate for zinc finger 288 to bind via its zinc finger domain. Now, if that's true, if we deplete MRE11, we should see less ZNF288 recruited and binding to the chromatin. And that's exactly what we see here. So here, we're after ionizing radiation, we see zinc finger 288 on the chromatin, but in the absence of MRE11, that completely abolishes this. So MRE11, we know, is functioning upstream of ZNF288 and creating, we think, a single-stranded DNA substrate for 288 to bind. What then happens is there's a switch from resection initiation to resection extension, which creates that long single-stranded DNA overhang. And it's known that this process is catalyzed by helicases and nucleases, but exactly how these enzymes are regulated in this context, it remains an outstanding question in the field. So we wondered whether ZNF288 could be involved in this regulation. So we simply asked, well, does ZNF288 interact with any of these key enzymes? And quite excitingly, we saw that 288 interacts directly with the helicase Bloom and the nuclease DNA2. So that puts ZNF288 within the Bloom DNA2 complex. Consistent with this, when we deplete ZNF288, we see less recruitment of this complex to a site-specific DNA double-strand break as well. And what we did finally was we teamed up with a lab in Switzerland, the lab of Petr Čeka, who has developed these very beautiful in vitro assays where he's able to assess the end resection activity of the Bloom DNA2 complex in a completely in vitro system. And here what they've done is they've added recombinant ZNF288 into this system, and they were able to see that this actually directly enhances the enzymatic activity of the Bloom DNA2 complex in a completely in vitro system, confirming what we've seen in cells. So our current working model then is that when we get a DNA double-strand break, resection is initiated by MRE11, and this creates a short section of single-stranded DNA which zinc finger 288 binds to via its zinc finger domain. And through interaction with DNA2 and Bloom, we think that enhances the recruitment of this enzymatic complex to the double-strand break site, where it can promote resection extension and create the most permissible environment for homologous recombination and DNA repair. So we're quite excited by this from a basic science point of view, where we think it's quite a neat regulatory mechanism, but we also think there is potentially an interesting translational and clinical component to this as well. So many end resection factors and end resection genes have been shown to either be mutated or deleted in human genome instability syndromes, including Bloom and DNA2. And a classic feature of many of these syndromes is microcephaly, which is the abnormally small brain development. So we reasoned, well, if ZNF288 really is a new critical DNA end resection factor, then perhaps it may too be involved in a human genome instability-like syndrome. And this is when we came across this 22q11.2 deletion syndrome. And this is the genomic loci where ZNF288 resides. And when we started looking in a little bit more detail at the clinical features in these patients, we saw things like immunodeficiency, microcephaly, short stature and growth defects, global developmental delay, and potentially even an increased risk of malignancy as well, which are all classic genome instability-like features. So 22q11 deletion syndrome is a bit of an umbrella term for a range of different human developmental syndromes, which are categorized by the size of the deletion. So the most common deletion is this 3 megabase deletion, this LCR22A to D region, and this is known as the DiGeorge region. The clinical features in these patients are relatively mild, and microcephaly is quite rare in less than 20% of these patients. However, there's this 1 megabase deletion, this D to E region, and this is where ZNF288 resides. And the clinical features in these patients is much more severe, and microcephaly is present in almost 60% of these patients. So we wondered whether the increased severity may be underpinned by increased genomic instability potentially. So what we did is we teamed up with the Children's Hospital of Philadelphia, who have the largest cohort of these patients in the world, and we were able to get some patient cell lines. So patient 1 has this DiGeorge deletion, which doesn't include ZNF288. So we're using this as a negative control essentially in our cells. And then we have 2 patients with the D to E deletion. So the first thing we did is confirm the haploinsufficiency of ZNF288 by real-time PCR. And then we simply asked, is there any evidence of increased DNA damage or genome instability in patients with this D to E deletion? And what we could see is that there's a significant increase in the number of overlapping gamma H2X and 53 BP1 foci in these patient cell lines, compared to patient 1, which has 2 intact copies of this D to E region. Now, to confirm that it was specific to ZNF288, what we then did is we used nucleofection to reintroduce wild-type zinc finger 288 into these patient cells. And when we successfully did that, we were able to almost completely rescue the DNA damage and genomic instability in these patient cell lines, confirming that haploinsufficiency of ZNF288 was likely impairing DNA damage and driving genomic instability in these patient cells. So I'll just finish up by reiterating our current working model, where we think ZNF288 is critical for the process of resection extension and creating the most permissible environment for homologous recombination repair. And we really think the take-home message here is in patients that lack sufficient quantities of this critical DNA and resection factor, zinc finger 288, we think it's impairing DNA damage repair that's driving genomic instability. And we think that that could in part at least explain some of the clinical features potentially that are seen in patients with this 22q11.2 distal deletion syndrome. And with that, I'd just like to finish up by acknowledging all of the people in my postdoc lab, which was Raul Mostoslavsky's lab over at the MGH Cancer Center, all of the people who have funded the postdoc, my postdoc, and of course, the people that are funding my lab here at BU now. So I started my lab in January. We're based over at 670 Albany Street, and this is my lab here that so we're just getting going. So I'm happy to collaborate with people, and yeah, if anyone has any questions, I'm happy to answer them. Thank you very much.
Any questions from the audience?
I have a question. Is the interaction with Bloom BTR dependent, or is it Bloom exclusive?
We haven't, well, so we haven't seen other components of the BTR complex, but we need to specifically check that. Yeah, and one of the things we're also interested in is looking to see if there is any, how important it is in the telomere as well. We haven't, because some of the clinical features that we see in these patients look like teleopathies. Yeah, very, very similar. So I've been to a few conferences where people have asked me, have you looked at telomeres? We haven't yet, but it's something we absolutely, it's on the list of things to do. So
we can help.
We should definitely talk for sure.
And I have a question. Can you overexpress ZNF280? Do cells tolerate it?
They don't love it. So
it's tightly regulated.
Yeah. So if we, and it's really interesting because the basal expression level is quite low in cells in general. It's quite low. Overexpressing it, the cells they're not particularly happy. But what's quite interesting in certain cancer contexts, it seems as though overexpression, at least in the context of colorectal cancer, correlates with chemoradiotherapy resistance. So one of the things we're looking at in the lab now is to see if the mechanism we've seen here with end resection and DNA repair, whether that holds true in colorectal cancer cells that overexpress it. And maybe that chemoradiotherapy resistance is through a similar mechanism here, but kind of the opposite of what we see in the developmental syndrome. So it's one of the things we're looking at now. So it's very tightly regulated, and it seems as though there's misregulation both in developmental disease and also certain cancer contexts as well. So it's a very interesting gene. Yeah.
Any other questions from the audience? Last chance. If not, let's thank our new faculty speakers, Dr. Clark, Dr. Ho, Dr. Isaac, and Dr. Muse for some really interesting science. I encourage all of you to reach out to them to foster and set up new collaborations. As we conclude this 16th GSI Symposium, I want to thank all of you audience members for attending. I'll be sending out a survey to all of you to give us some feedback. We're always receptive to things we can improve in the GSI. Continue to look out for events happening within the GSI. I also want to thank all the trainees, poster presenters, and lightning talk speakers for their excellent science. Us faculty that judged the posters. Also my colleagues, co-director Dr. Ala Grishak and Dr. Rachel Flynn, as well as our admin, Sabina Dahal, somewhere here. She really helped me out a lot in organizing this symposium. We'll be working on deciding who are the grand prize winners from the lightning talks and the poster sessions later. And thank you all again for attending, and hope to see you all again next year at this great event. Thank you again.